Hey, curious minds! Welcome back to the Curious Scholarrette, where we take complex science and make it beautifully simple. Today's story is one of hope, ingenuity, and maybe, just maybe, a giant leap toward curing HIV. We're diving into an incredible piece of research just published in Nature Communications, where scientists successfully used mRNA and lipid nanoparticles, yes, like the ones in COVID vaccines, to wake up the dormant HIV hiding in the body. If you've ever wondered why HIV can't be cured or what it might take to beat it for good, this video is for you. Let's start with the basics. HIV, the human immunodeficiency virus, is a master of disguise. Once it infects a person, it integrates its genetic material into the DNA of certain immune cells, mainly the resting CD4 T cells. These infected cells can remain in a dormant state for years, even decades, without producing new viruses. This is what scientists call the latent reservoir. Why is this a problem? Because antiretroviral therapy, ART, only targets active replicating HIV. The virus hiding in these latent reservoirs escapes treatment and reawakens if ART is stopped. So despite enormous advances in HIV treatment, there is still no cure. And this is where today's research becomes groundbreaking. In this new study, researchers developed an innovative solution, a lipid nanoparticle-based mRNA delivery system called LNP X. If the term sounds familiar, that's because lipid nanoparticles are the same delivery vehicles used in the COVID-19 mRNA vaccines. But here's the twist. Instead of delivering instructions to make spike proteins, this system delivers mRNA that reactivates latent HIV. In other words, it forces the virus out of hiding so the immune system, or future therapies, can target it. To test their idea, researchers engineered a synthetic mRNA that encoded a protein capable of waking up latent HIV. They packed this mRNA inside the LNP X nanoparticles and tested them in lab-grown resting CD4 T cells, the very cells that host hidden HIV. Here's what made this effort remarkable. Most delivery systems fail to enter resting T cells because these cells are, well, quiet. But LNP X was able to sneak in and deliver the payload without activating the cells or causing widespread immune activation, a major concern in HIV cure research. They confirmed that the LNP X particles were taken up by the cells, that the mRNA was translated into protein, and crucially, that this protein reactivated HIV. The results were jaw-dropping. First, LNP X achieved high mRNA delivery efficiency, up to 60% in some T-cell models. Second, the delivered mRNA successfully reversed latency, meaning the dormant HIV became visible. Third, this was achieved without toxic side effects or triggering unwanted T-cell activation. Translation: the researchers woke up the sleeping virus without sounding a global immune alarm. This is a critical step toward what's known as the shock and kill approach, where the virus is first shocked out of hiding and then killed off by the immune system or additional therapies. This discovery could be a pivotal turning point in HIV cure research. For over 40 million people living with HIV, a cure has been a distant dream. This research brings us closer to making that dream real. Not just managing HIV, but eliminating it from the body. It opens the door to therapies that are safe, precise, and capable of reaching the virus where it hides. And beyond HIV, this technology could also be adapted to other diseases involving latent infections, like herpes or even some cancers. So, is this the silver bullet for HIV? Not yet. But it's a massive leap in the right direction. If you found this as fascinating as I did, give it a thumbs up, share it with your curious friends, and don't forget to subscribe to the Curious Scholarrette for more science made simple. Let's keep questioning, learning, and imagining a healthier future, one discovery at a time. Until next time, stay curious.